Before we begin talking about using the information in DNA to make proteins, let's review DNA versus RNA. First of all, remember DNA is deoxyribonucleic acid. RNA stands for ribonucleic acid. DNA is found within the nucleus, RNA in the nucleus, and the cytoplasm. DNA is a double-stranded helix held together by hydrogen bonds. RNA, single strands. Now, both DNA and RNA are made up of nucleotide monomers. Deoxyribonucleotide monomers, remember, mono, one, mer, piece, or unit. So the DNA monomers are adenine, thymine, cytosine, and guanine. A always complementary base pairs with T. C always complementary base pairs with G. In RNA, we don't have thymine. We replace it with U, which stands for uracil. So with ribonucleic acid, RNA, the monomers are adenine, which pairs with uracil, and cytosine and guanine still pair together. The enzymes used by DNA polymerase, RNA polymerase. Yep, sometimes it's just that easy. Now, 3 base pairs of DNA form a triplet. 3 base pairs with RNA make up something we call a codon. We have 3 types of RNA, and we'll look at each one of these. Messenger RNA, ribosomal RNA, and transfer RNA. Now remember, DNA holds the instructions for making proteins. In little sections of DNA, we call a gene. I want you to think of it this way. All of your DNA is like a cookbook. What do you find in a cookbook? Well, if you want to make a chocolate cake, you open it to the page that says chocolate cake, and it tells you exactly what ingredients to put in, how much of each to put in, when to put them in, what to do with it, how to bake the cake, what temperature, the whole 9 yards, right? It tells you exactly how to make that cake. That's what we call a recipe. Well, what if we want to make a protein? We need a recipe to make that protein in our cells as well. So your DNA is the cookbook. Each individual gene is a protein, or each individual gene is the recipe for how to make a protein. All right, so protein synthesis. Synthesize means to make or form. So there are 2 big steps that we'll examine. First is transcription, which occurs in the nucleus of the cell. And it's the process where a segment of DNA is read, like you would read the recipe, in order to make a new product. Well, in this, we're going to make RNA, which is then going to be translated in step number 2 to make a protein. So again, your transcription happens in the nucleus because that's where the DNA is. Transcription, think of it to copy, to write something down. So if Granny's cookbook could not leave Granny's kitchen, but you wanted that recipe for chocolate cake, you would have to write it down or transcribe that information and then take it to your kitchen where you would translate that into a chocolate cake. So your kitchen would be the ribosome, which is in the cytosol of the cell, and that's where we take that messenger RNA that was carrying the DNA message, we take it and translate that into a protein. Now, as we start to use that DNA information to assemble a protein, transcription, remember, is the first step. So the DNA will serve as the template. It's the recipe in the cookbook. DNA helicase is an enzyme that separates the 2 strands. So remember the double-stranded helix? Helicase breaks that apart or separates the 2 strands. Then RNA polymerase is going to add nucleotides to form messenger RNA. So polymerase forms a polymer. Remember, polymer, many pieces. So your helicase splits the DNA. Polymerase is going to add the nucleotides. Now, there are 3 types of RNA. We mentioned those a few minutes ago. We'll talk first about messenger RNA and its role in taking the message from the DNA so we can turn it into a protein. There are 3 events that have to happen during transcription, okay? Initiation, elongation, and termination. The example your book uses is spot on, so again, I'll give you that one. So transcription, you can compare it to writing down that recipe, like I said a while ago. But the DNA, of course, is in the cookbook. The gene is the recipe. So you open the cookbook, that's initiation. You write down the recipe, that's elongation. And then you close the book when you're done, that's termination. Now we'll talk a little bit more about that. Let's begin with initiation. So DNA helicase unzips the DNA. Remember, it's usually a double-stranded helix. So the helicase unwinds it, opens it up so that the gene is exposed. Now, once that is done, RNA attaches to the DNA. RNA polymerase attaches to the DNA. Polymerase is the enzyme that's going to move along the strand of DNA until it reaches something called a promoter region, which is the start point for transcription or getting that information. Now, because RNA is single-stranded, only one strand of the DNA needs to be copied. So the DNA strand that's going to be transcribed to form the RNA is called the template strand, and the other one that is not copied is called the coding strand. So remember, with initiation, find the promoter, get ready to copy. The second part of transcription is elongation, where nucleotides that are found within the nucleus are going to be base paired just the same way we saw with base pairing, A's, T's, C's, and G's. We're going to base pair the A's, T's, C's, and G's of DNA with A's, U, G's, and C's to make RNA. So the RNA polymerase is going to move down the strand of DNA until the entire gene is transcribed. So hydrogen bonds are going to form between the template strand of DNA and the new RNA nucleotides that are coming in to complementary base pair. Again, remember, base pairing involves forming hydrogen bonds between the nucleotides. So where we have a T in DNA, an RNA nucleotide of adenine comes in because T is still going to bind with A. But remember, we don't have T's in RNA. So anywhere there is an A, instead of getting a T like you would for DNA, we're going to put a uracil instead. So we're creating a strand of messenger RNA made up of nucleotides based on the template strand of DNA. And we do that until those base pairs will continue to form. The nucleotides come in until we reach the end of that gene. Basically, think of it this way. When you get to the end of the recipe, you stop writing down the information. You can see that happening here in this image. So the big purple thing here is our RNA polymerase. That is the enzyme that is slowly moving down the strand, the template strand of the DNA. So here's our template strand, and here's that RNA that is forming by these ribonucleotides coming in and forming hydrogen bonds with the DNA nucleotides on the template. So when the RNA polymerase reaches the end of that gene, that's called the terminal end of that gene, then the polymerase will be released from the DNA. Hydrogen bonds are broken between the DNA strand and that new messenger RNA that we made. The DNA is going to go back together and rewind into a double helix, and the new messenger RNA now has that recipe that we copied from the DNA. so that we can turn that into a protein, such as insulin. Here you have all three steps that we just described, making up the first part of protein synthesis. Remember, transcription is the first step, and it has initiation, let's start, elongate, add more nucleotides, and terminate, we're done, end of the recipe. Okay, so all three of these steps must occur before we can now translate that information into a protein. Before that messenger RNA leaves the nucleus and goes out to find a ribosome so we can turn it into a protein, modifications will typically occur to that strand of messenger RNA. There are several ways to modify that messenger RNA, but one of the ones that I just wanted to point out was something called removing introns. So introns are non-coding regions. Basically, it's sort of like the parts of a recipe that really don't mean anything. So like if Granny told you in her chocolate cake recipe that I learned how to make this chocolate cake from Aunt Sue. Well, Aunt Sue and the information about where Granny made it doesn't have anything to do with how much cocoa and how much sugar and how much flour you put into that chocolate cake, right? So it has nothing to do with the recipe. So that's sort of what introns are. It's little pieces of RNA that don't make any difference because they don't tell us how to make the protein. So introns are removed, and then the exons, which are the leftover parts, are spliced back together. So that modified now and mature messenger RNA will exit the nucleus through a nuclear pore, and once it leaves, it will find its way to a ribosome where translation, or step 2, can occur. Since we're talking about making proteins, let me remind you, proteins are made by amino acids forming something called peptide bonds between each other. Think about a pearl necklace. Each individual pearl is strung together with other pearls by the little string, right? So each pearl would be an amino acid. The peptide bonds would be the string that holds them together. So how does all of this work when we have to put amino acids together in a particular order to make a particular protein? Well, this codon chart will help you understand that very easily. So remember when we unwound that DNA to expose the gene, we had 2 strands now of A's, T's, C's, and G's. And then we transcribed that into 1 strand of RNA, A's, U's, C's, and G's. Okay? Now, I mentioned on one of the previous slides that each set of 3 RNA nucleotides is called a codon. That codon simply is those 3 nucleotides together, and that determines which amino acid is brought in to form peptide bonds to form that protein chain. So let's look at this table. If you notice here on the far left side, this says first letter, there's U, C, A, G. So let's go with A. So first letter, A. Everything in this row, the first letter is an A. Second letter, let's go with U. So everything in this column, the second letter is a U. Third letter, okay, so your third letter, everything in a row across will be whatever is listed here. So let's stick with this one. We'll go with G. So look, third letter G, 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 G. Okay, so A, U, G means that we need a methionine. Methionine is always going to be the first amino acid added to a chain. So our mRNA starts with AUG. Now, what if the next set of 3 nucleotides, the next set, okay, the next codon, what if it is CCU? Well, what do? We go up here and find C. All of those are C's. Come to the second letter, C. That's this column. Then U, third letter, CCU means the next amino acid would be a proline. And we continue using this layout to add specific amino acids until we reach the end of that messenger RNA, and then we have something called a stop codon. So you see these three right here? Look what the amino acid is. Oh, stop? No, stop is not an amino acid. Stop means there's not an amino acid that this codes for. So what do? You can't add another amino acid. Guess you're done. Translation is making a new protein, and it takes place at ribosomes in the cytoplasm or in the cytosol, the liquid of the cell. The RNA, the messenger RNA, is moved through a ribosome and the information is read. Now, the code in the nucleotide sequence of messenger RNA is translated, which simply means it's used to detect the way we bring those amino acids in and hook them together to form a long strand of amino acids. Remember, that was our first level of structure when we learned about proteins back in the previous chapters. So in order for translation to occur, we need the ribosomes, that's where we make proteins. We need the messenger RNA. We need transfer RNA. And we need amino acids. Again, your book gives a really good example of this. So the components that are required for translation, think of it like a chef in the kitchen getting ready to cook your favorite dish. So the ribosome is the kitchen. The messenger RNA, we talked about that already. That's our recipe, right? The tRNAs, transfer RNAs, are the chef assistants. So that's your sous chef and your other cooks in the kitchen. You need the amino acids, which are the ingredients, and then the protein would be the dish we cook from the recipe. So the transfer RNA assistants are going to be responsible for bringing in the right ingredients, bringing the amino acids to the kitchen. Now remember, the kitchen is the ribosome. And then, once all that is done, we've made our recipe. We made a protein. So let's look at this image for just a moment. Here's our messenger RNA, okay? Here is one way of looking at transfer RNA and another. It's really just a molecule that is going to bring in amino acids. That's its job, okay? So at one end, we have something called an anticodon. Anti, think of it like opposite. So it's going to pair with a codon on messenger RNA. Now remember, we said on the previous slide that the start codon is always going to be AUG because it codes for methionine. So what would complementary base pair with AUG? Think about it for a moment. What does A bind with? It's RNA, so A goes with U goes with A, G goes with C. So in order for a transfer RNA, in order for our transfer RNA to pair with this, it would have to have a U, an A, and a C. And if you look right here, U, A, C. So that base pairs with this and bam, brings in an amino acid. And that's exactly how it works. Each codon, each set of 3, tells a particular transfer RNA to bring an amino acid to add to the chain. Just a brief look at the ribosomes. Ribosomes are made up of protein and ribosomal RNA, which that's what we call rRNA. So there's a large subunit, a small subunit. The ribosomal RNA acts as a catalyst to help assemble the amino acids into a protein. So remember, the ribosome is the protein making factory. Your messenger RNA, again, we've seen this. It carries the message from the DNA. It has the recipe written down, brings it out to the ribosome so we can read that message 3 base pairs at a time. Remember, 3 base pairs of messenger RNA is a codon. The start codon is always going to be where we begin adding amino acids. It's where we start reading the recipe. That will always be the AUG. And then our stop codon, remember, that's when we don't have an anticodon on the transfer RNA that matches it. So there's no tRNA to come in. Therefore, the protein stops adding amino acids at the stop codon. Another quick look at the transfer RNA. Remember, this is what will transfer amino acids to that growing polypeptide chain. So in its simplified version, the tRNA molecule looks like a clover leaf. It has two distinctive and significant regions, the amino acid acceptor end and the anticodon. So the amino acid acceptor end is where a particular amino acid will attach to that tRNA. The anticodon is a specific sequence of 3 nucleotides for each transfer RNA. Now remember, the anticodon will complementary base pair with the codon on the messenger RNA. And that's how the correct amino acid gets brought in. The DNA had the information. The messenger RNA copied it and brought it out, and then that messenger RNA has sequences of 3 nucleotides we call codons. Anticodons that match that bring in particular amino acids. Just like we had initiation, elongation, and termination as the 3 steps of transcription, we have the same 3 steps in translation. So initiation is simply getting everything together. So the ribosomal subunits, the large and the small, will form a complex with the messenger RNA and the transfer RNA. Base pairing then between the transfer RNA and the start codon of the messenger RNA, that happens next. And remember that start codon is always AUG, so methionine is always that first amino acid. Now, we've initiated the process, we're ready to go. Elongation, very simple. The rest of the transfer RNAs bring in their amino acids and peptide bonds form between neighboring amino acids, forming a linear strand of amino acids. During that elongation process, the ribosome is continuously moving down that string of messenger RNA, 3 nucleotides at a time, until it reaches a stop codon. Now remember, a stop codon is the codon that does not have a matching transfer RNA. So if there's no anticodon on a transfer to match that messenger RNA codon, then nothing else gets brought in. Okay, so that's your stop codon. The ribosome breaks apart. The new string of amino acids and unfolded protein is released. Now, a single strand of messenger RNA can be read or translated by more than one ribosome at a time, and that allows many copies of a particular protein to be made at once, and we call that messenger RNA is polyribosomal, which simply means it has a lot of ribosomes. Many, poly means many, so we're making many copies of it all at once. This slide just takes you through the entire process of translation from initiation with that start codon, the elongation of adding subsequent amino acids, forming peptide bonds between them, then the transfer RNA, it's dropped off its amino acid, it goes off and reloads so it can bring back more, and another one comes in, and then that amino acid will form a peptide bond with these two, and then this transfer RNA will fly off and go get another amino acid, reload, ready for the next time. Termination, of course, means to stop. So when we reach the stop codon, there is no transfer RNA to match that codon. So we stop, and now that protein chain, that primary sequence of amino acids of that protein has been completed. A disorder associated with this that's really easy to understand is sickle cell anemia. Now, sickle cell anemia is a condition where your red blood cells can't carry enough oxygen. So red blood cells have molecules of hemoglobin within them, and hemoglobin binds to oxygen. Hemoglobin is a protein. Remember, we talked about this as our example of a quaternary protein, where we have 4 amino acid chains all hooked together. So we have 2 alpha chains and 2 beta chains. But somewhere along the way, something got messed up in that sequence of A's, T's, C's, and G's, or A's, T's, C's, and U's. So instead of coding for a particular amino acid, let's see, it was supposed to be a leucine. And so the codon was CCU, but there was a mutation, and instead of CCU and a leucine, we had C or CUU and a leucine. So instead of CUU, we have CCU, and now the transfer RNA brings in a proline instead of a leucine. Well, that changed our sequence of amino acids, and that can cause the protein not to fold properly. Well, if a protein doesn't fold properly, sometimes it can't do its job. And if it can't do its job and it's hemoglobin, then of course that results in a hypoxic or low oxygen condition. And it all started because the recipe was bad. So it's a genetic disease. Imagine if Granny's recipe for chocolate cake had a cup and a half of salt instead of a cup and a half of sugar. Then you would put in the wrong ingredients and it sure wouldn't be a good cake. So that's what happens with a lot of our genetic diseases and disorders. We have the wrong amino acids coming in to make that chain.